Hi, good morning to all. I am Ancy, Assistant Professor, Department of Biomedical Engineering from Emmanuel Sarjeje College of Engineering, Nattalam. Today we are going to discuss about blood cell counter. Blood cell counter is a device used to count the number of blood cells including RBC, WBC and platelets. Blood cell counters, also known as automated hematology analyzer, are devices used to perform complete blood count test by quantifying the blood cell parameters with verification methods guided by the clinical and laboratory standards institute or the International Committee for Standardization in Hematology. Two basic cell counting technology is used. First one is electrical impedance. It was the only automated cell counting method. Second one is light scatter cell counting. First one is electrical impedance cell counting. The principle: in an electrolyte solution which conduct electricity, blood cells count out by electrical impedance are diluted. The electrical current passes through the opening of the measuring device in the electrolyte solution from one electrode to another. In this diagram, external electrode, internal electrode, two types of electrode used, then aperture tube. These are the blood cells diluting fluid. Then blood cells are the bad conductor of electricity. Non-conducting cell create an impedance or disruption in the electrical circuit. When a cell suspended in the electrolyte solution passes through the aperture, the impedance of the electric circuit induces a pulse. Those pulses are known to be cells. The impedance size is proportional to the size of the cell. Then next one is light scatter cell counting. The tiny channel in a machine called a light scatter cell counter guide blood cells in a single line. The laser or light beam shines on them, and when a cell is hit by the light, it scatters in different directions depend on what kind of cell it is. This is because of something called the cell refractive index, which is based on the shell shapes and size. The machine measures how much the light the cell absorbs and scatters to determine the type of cell. These are the light scatter cell counting. The light source by using the photodetector and light source, it can the cell dilution, and photodetector should detect. Then sheath stream, cell stream, flow cell. The laser light employed in the light scattered cell count is unique. It is just one wavelength, so travels in a straight line. As a result, is more exact than other forms of light, such as tungsten halogen light beams. The laser beam is very effective in diagnostic hematology because it may produce highly accurate scatter patterns that can be utilized to distinguish various types of blood cells. Then red cell measurement. Red cells can be measured by aperture impedance light scattering techniques using laser LED or tungsten or a combination of two technology. First one is reticulocyte and nucleated RBC measurement. The reticulocyte on the device cell counter also have an evaluation. Many devices are RNA fluorescent dyes with laser light to test reticulate and related parameters. White cell measurement. There are white cell measurement several methods for counting white blood cells, each with own set of advantage and disadvantage. A sheath flow based approach is one unique method of counting them. Depending on the manufacturer, the sensing area can employ various forms of signals such as conductance or impedance, and the sensors can look at various things such as light scatters or fluorescence. This is platelet count. Platelet count by using the hematology analyzer. This is numerous approaches including impedance, light scatter, a combination of two, and flow cytometry with fluorescence can be used to count platelets and measure their size. Platelet count based on impedance is used by certain equipment to calculate the amount of platelets in a sample. For samples with platelets more than 20 femtoliters, curve fitting software is employed to increase accuracy, extending the platelet count range 70 over the 35 floor limit. Thank you.